Well, we are circling around the old story. We are still trying to understand the biology behind immune checkpoint inhibition. A lot of it is known, but a lot of it is still to be identified. One particular aspect, which is very critical and very pressing, is to find the biomarkers that can way better produce some kind of clinical algorithms that can predict which patients would benefit from these treatments and which will not. With the knowledge in biology, as well as the clinical setting, we are understanding some new information that is being generated in these studies. While we have a better leverage of the data in the tumor setting, meaning in what's happening in the cancer, particle in the cancer mass, in the tumors, relatively less information is known about how all that environment in the tumor is influenced by pre-existing host immunity. In our efforts that we have developed in both metastatic as well as adjuvant setting, we are trying to understand how the pre-existing host immunity that is usually determined by the composition of the blood cells, circulating blood cells, may give us some kind of information that can somehow let us know which patients before the treatment would benefit from these drugs. We are learning that the composition of the peripheral blood lymphocytes is critical. We can understand their phenotypic distribution. We can understand perhaps the type of blood cells that is circulating. That may be potentially important in the anti-tumor immunity. However, very little knowledge is generated about the genomic or transcriptomic regulation of these blood cells. We are developing a lot of technologies such as single-cell analysis, maybe more high degree based immunophenotyping, very deep immunophenotyping methods. And this information is very critical. However, what is really important is going to the basic molecular level, understanding what kind of genomic imprints are actually pre-existing in the different blood cells in our circulation, and how those genetic imprints somehow shape up the host immunity. These efforts that we have been developing in the context of adjuvant setting as well as metastatic setting in melanoma bring up that information on the resolution of the single cell. We are trying to identify all genomic and epigenetic imprints in the peripheral blood cells and potentially identify the specific phenotypically interesting clones that can somehow explain which of these cells would eventually circulate to the tumor and which of these cells would really do will mount the very efficient anti-tumor response. We are identifying novel transcriptional regulatory networks in these peripheral blood cells that are composed of different layers of genomic information, such as high-resolution transcriptomic profiling, usually through the RNA-seq analysis, single-cell RNA-seq analysis on the single-cell level resolution, most importantly, some epigenetic regulatory pathways as well, using the more complex assessment of the open chromatin sites through the methods such as ATAC-seq analysis, which is also available now on the single-cell resolution. And putting all these multi-omics, genomic, transcriptomic, epigenetic information together in these big algorithms will allow us to generate a complex genomic regulatory networks in a particular cell types in our circulation that we can then interrogate backwards in the tumor and find the correlation. This will have enormous clinical implication, not only substituting the analysis in the tumor, but also giving us better analytical tools to supplement this very high degree difficult genomic analysis and interrogation in the tumor specimens to just simple test in the blood, which will potentially be able to find these surrogates in a much better resolution. So these methods are constantly evolving. We are on the brink of the extremely interesting and exciting era of a lot of next generation technologies, including artificial intelligence that will be very helpful in this context. But collectively, all of this data will be accumulated, hopefully, in a large repositories as part of clinical trials and beyond, and we'll be able to develop way better predictive algorithms, now incorporating all the information from peripheral blood.